Research that sponsored this talk today, this symposium. And so I'll direct your attention to just a little bit of past history about this organization that is so wonderful in its support of medical students and other students in training to try to bring them into basic and translational medical research. We have so few venues for that, to expose students to that, and we're so proud to do that. Last week I was at the AFMR conference in Philadelphia, and many students got their first chance to be on the podium, and it's so exciting to be able to see that and be part of that and build enthusiasm for this, because we need our future scientists. We need our people of tomorrow to keep trying to innovate. We have distinguished past presidents. We have a rich history of Nobel Prize winners, and we have wonderful committee of dedicated physician scientists working to keep the organization going. So I just wanted to point that out and let you know about us, make you aware of us. We're online, we're on Twitter and Facebook and everywhere. So I hope you'll look us up. We have a wonderful journal, the Journal of Investigative Medicine, and a high-impact journal. We have, as I said, many meetings that really give young people exposure to science and a chance to be presenters themselves to get ready for national meetings. And so I just want to give you a little background there. Okay. So welcome, everybody. It is my pleasure and honor to be here. I'm Allison Reiss, coming here from New York, if you can't tell by the accent, and I'm chairing your symposium today. And I think you'll find that this is a very timely and exciting topic, and not just that, but a topic that has so much importance and so much potential of impact. Alzheimer's disease, such a terrible disorder, so prevalent. We're so helpless in the face of it, and we need to do something. And that is the title of today's symposium, Alzheimer's disease, many failed trials. So where do we go from here? And today I've brought to you some of the most distinguished people with out-of-the-box thinking on Alzheimer's disease, who are going to talk to you about what they think, what they're doing, and how we can move the needle on this, because we're really failing in many ways to do that right now, and we need to make some changes. So Mark Stecker, professor at UCSF, and Ben Willozin, professor at Boston University, both MD, PhDs with incredible track records and backgrounds in this field. And we're going to start with Dr. Stecker, so I welcome Dr. Mark Stecker.
Thank you, Allison. And I just have to put in the plug that AFMR really is a great group, and they really do bring all these students together. Now, how do I get this now that I don't have the, wait, here we go, I got the cursor. I can just shrink this now and do this. Got it, hold on, we got it. I think we got it, there it is. Okay, but I do have to put in my tab for the AFMR. It's a really great organization, and it really does bring students that don't have a chance otherwise to really do some things. So I wanted to talk about, as a clinician, I'm more talking about the negative aspects of trials and suggesting some ways forward. And I think we can do it, but we have to change the paradigm. So I'm going to talk about issues in clinical trials because we have difficulty diagnosing dementia. I'm going to have issues because talk about the issues regarding lack of funding per the demographic. Some of the characteristics of the clinical trials in dementia that are going on right now and how they may not be optimal. Some of the financial perspectives on how clinical trials are done in dementia. Some of the issues with animal models versus human trials. And then talk about some ways forward that we can all do. So we start with the question, what is dementia? And as always, we turn to manuals and books. And the DSM-5, the Psychiatry Diagnosis Manual, gives us this definition of dementia. Significant cognitive decline. How significant? What number do I use? Don't know. It's a qualitative thing. Problems that interfere with independence in everyday living. How much? How accurate is that? The disorder is not something else. you can see that that's a great clinical diagnosis. But to give you pathophysiology, you're not really close. It's too diffuse. And to make it worse, dementia is not a single disease. There are many different diseases that produce dementia. We know that Alzheimer's disease is the most common of those, but vascular dementia is not uncommon, and a mixed picture of vascular plus Alzheimer's is pretty common. Lewy body dementia is not uncommon, Parkinson's dementia, and frontotemporal dementia, all of which have different pathophysiologies. Now, they all have different pathophysiologies. If you can't tell them apart, how will a drug that works for one show up in a clinical trial? Of course, there's less common forms of dementia: hypothyroidism, alcoholism, normal pressure hydrocephalus, B12 deficiency. These are all other things. So there's a lot of things that go into that diagnosis of dementia, and it's hard to be sure. So of course, we go back to a manual that tells us what to do. So we then look at the DSM and it says, what's Alzheimer's disease? Well, that's the definition. Insidious in onset. How insidious? How slow? How gradual? Clear-cut history. How clear? So all this is great for a clinician. If I'm going to decide to give you a pill, I maybe can do it on the basis of this. That helps me a lot. But if I'm doing a clinical trial and I need to know whether 25% people get better or 20% of people get better, I'm not going to be able to answer that question with this kind of diagnosis. And so pathology and biochemistry, the only ways to be absolutely be sure. We know that when Alzheimer discovered Alzheimer's disease, he found in the extracellular media amyloid plaques. And we know that tau is the intracellular accumulation. And in studies that have been done, that 83% of patients that are diagnosed with Alzheimer's disease had the pathologic changes. And sensitivity to the clinical diagnosis ranged between 70 and 87%, and specificity was really poor at 44 to 71% in this study. So we don't have a great way to make that diagnosis in life. Okay, that's different from other diseases we're discussing. So just think about this. Again, if 50% of the patients in your cohort for a clinical trial had something other than Alzheimer's disease, and you had the drug to cure the Alzheimer's disease, it'd be really hard for that drug to show up positive in a trial. So it already goes against this trial being positive. And it's almost as crazy as Hieronymus Bosch extracting the stone of madness from someone's head. Because in cancer, we don't give you chemotherapy until we've biopsied something and we know exactly what's wrong. Infection, until we grow out the bacteria. Diabetes, we can measure things. We measure glucose. Heart disease, we have good measures. In dementia, we don't. So that's one of the things that we have a problem with in designing drugs and treating this. It's a heterogeneous group of diseases that are hard to make that diagnosis. In vivo chemical diagnosis, we know that we've now been able to classify the different dimensions by what accumulates. If tau accumulates intracellularly, or if alpha-synuclein accumulates intracellular, or TDP-43 accumulates intracellular, that's great. But even though we have PET scans where we can look at things and find deposition of tau and amyloid, we know that normal patients can have amyloid and tau deposition. So it's still not 100% marker for what's going on. So we have diagnostic uncertainty, but we also have phenotypic variability because there are a ton of things that change and modify the course of dementia in everybody. So it's like we're playing children's games from Bruegel. Everybody's playing a different game here because we're all individual and different. And if you just look at the risk factors, right? Education, mental activity change the time course. Age affects this. Race, ApoE4, family history, type 2 diabetes, obesity, all change this. So not only is it a slowly progressive disease that's hard to make the diagnosis correctly, it's also hard to say what the time course is going to be for an individual group of people. So it makes it really hard. So we have to think about that when you say trials don't work. Let's just look for a second about what the societal cost of dementia is and how much we invest in that. So say between 2 and 5 million people have dementia, and we know that the risk of dementia goes up as you get older. It's the 6th leading cause of death, but the only one for which we have no disease-modifying therapy. So if you look at these estimates here, it's $300 billion a year by 2020, next year, assumed to be caused be related to dementia. Now let's look at some other comparison numbers. NIH, 37 billion, 1/10 of that. NINDS at 1/100th of that. It's 30% of the entire Medicare, Medicaid budget. So it's a large amount of money. And the total funding is very small. So if you look at this, and again, Ben and other people would know better than I, but drug companies put a lot into R&D. They might put 10%. So that means if a drug costs $300 billion, if a disease costs $300 billion a year, I might want to put in $30 billion. But in fact, what we're putting in is about 0.6% of what this costs in research, instead of the 10 or 20% a successful group would put in to fixing a problem. And it's 9% of the US budget. Yet we're only putting in 0.6% of what it actually costs. So you need more money. There's no question about it. Let's just have a quick look and compare. You say, well, maybe it depends on the prevalence of the disease. So here's things this is all from clinicaltrials.gov. You see cancer here. It has 14,000 recruiting trials and prevalence of 15 million and roughly 0.92 recruiting studies per 1,000 people. Dementia is 0.12. It's not so dissimilar from some of the other diseases, but nowhere close to cancer. We'll talk about that in a second. So dementia is underfunded in regard to its effects on the economy. The number of recruiting trials could be a lot better, and it's not even close to that of cancer, because we had the war on cancer, and it made a huge difference in what we did. We said we can solve this problem. We've never said that we can solve dementia. We should be able to say that. We haven't. Once we give a positive statement, people will be more interested. Understanding the type of clinical trials is the next thing I want to talk about, and we're going to get back to the difference between a cancer clinical trial and a dementia clinical trial. So let's look again. This is all from January on clinicaltrials.gov. All of you can look that up and see what kind of trials are out there. So 46% of trials involved a drug, 7% involved devices, 16% involved behavioral modification, and about 40% had an industry sponsor, and only 13% of the funding came from the NIH. Okay? It's a little less than for cancer, but now we're going to see where a lot of the problems come from. What kind of clinical trials are recruiting? 15% imaging, diagnosis. Behavioral treatment, 15%. Drug therapy to modify the course of the disease, only 60%. So roughly half of the trials that are out there are trying to have a curative procedure. So that's pretty low. If you look at the trials, again, where they're distributed, we know the phase 1, the early phase trials, and the phase 4, the largest trials. For dementia, many of the trials are early here. They're phase 2 trials, whereas the phase 3 trials and phase 4 trials are really hard to come by because they fail in phase 2. And if you look at total recruitment on clinicaltrials.gov, there are 24,600 people registered by the government in dementia trials. There are 1,600,000 people in cancer clinical trials. There's 66 times as many people in cancer clinical trials as there are in dementia clinical trials. That makes a huge difference. We're going to talk about why. First of all, there's more funding for cancer research. We all know that. But for cancer, there's a clear individual benefit. Anyone who has cancer who's not being treated effectively by the drugs they're getting will go, there's a clinical trial out there, and I want to participate because that has a chance of making me better. Because new drugs have a track record of working in cancer. And you can get the advantage of getting other therapies for free. If you were taking 3 drugs and you participate in a trial, you may get one of those drugs for free. So the cancer patient drives a lot of this research. They want to participate. They know that they can be cured maybe with a new drug. The dementia patient doesn't have the cognitive ability to drive that. And it's, of course, hard to find and recruit dementia patients. So we need to change that paradigm. We need to give people hope in the dementia world, or at least get them something back. Okay. Now, we need to also make it easier for people to recruit patients into trials. We'll talk about it again. Change HIPAA, right? makes it very, very difficult. Establish registries, community outreach. I'll talk about the Department of Motor Vehicles in a second. If we can get a likely individual benefit for participating in trials, it's great. You know how hard it is to bring a dementia patient into a trial. The family has to work. It's a lot of effort to do that. If we can make that easier, it's great. So let's talk now. I'm going to go back one more time from clinicaltrials.gov. This is as of January, every drug that was being studied as it was registered in clinicaltrials.gov. And you see some drug company names here, but you see grapeseed extract. You see a lot of natural agents here. So if you summarize just what's being done as of January, 12 of those studies were herbal studies. Just because they were available and there's maybe some evidence, they're being studied. Amyloid and tau, just a few studies on amyloid and tau. Beta secretase, a couple of studies. Cell modulation, but there's things like phosphodiesterase, hormonal therapy, stem cells, of course the vaccines. So it's a variety of things, not all of which actually target what we think is important. So they're not all targeted on mainstream pathophysiology. Only 33% have anything to do with amyloid or tau. And so a lot of those are chosen out of convenience. It's easy to study an ancient Chinese herb. Is it going to work? Maybe. But we don't have the pathophysiology to back it up. Of course, you can determine a pathophysiology to make that work and do some studies. But, you know, if we're thinking in this direction, maybe we should all move in that direction. Maybe this is not right, but we need to keep thinking. Trials have not been effective, of course, in tau and amyloid, so that's another problem. And in the clinical trials, side effects are high because you're dealing with the elderly. Elderly have side effects on just about everything. Now, again, Ben will be able to give more information and be much more erudite about this, but a lot of the major drug companies are dropping officially out of the Alzheimer world, even though they may be sponsoring other things in the background. Because if you think about this, if I'm a drug company, if I give a drug to somebody with Alzheimer's disease who had it for 5 years, who has brain atrophy and loss of brain cells, they're not going to get better, right? I need to intervene in the early stage. Well, in the early stage, I need to know how quickly they're going to progress. What would the natural history be in this individual patient? And it may progress over years. So my clinical trial will take a while. That could take years to get a good clinical trial. Now, with a huge cost, $500 million to bring a drug to market, the drug company's got to recoup their investment by the end of the experiment, by the end of the experimental phase when they finally introduce it. If they get 20 years of patent life, it takes 10 years to do an experiment, they've lost half the potential value of that drug. They would much rather study a blood pressure pill. You give them the pill, measure the blood pressure tomorrow, and you got an answer, rather than treat Alzheimer's disease, wait 10 years, are they any better? That's a long, complex, expensive study. And again, patients will pay out of pocket for the cancer treatments, right? If you have a life-saving drug, they will pay for it. The dementia patients won't. So the drug companies know that and know that they can increase the prices for the cancer drugs because people will pay. So they know that they can get reimbursed for some of these things. We have to change that. So everyone else is going to talk more about this. We know that most therapies are based on amyloid and tau, and we know about the story of A-beta, which is part of the amyloid precursor protein, which is intracellular and broken out by beta secretase and gamma secretase. And it's the aggregation of A-beta extracellularly that's associated with disease. And if we just look at pathophysiology, I just want to show one example of this. This is a beautiful animal study on beta secretase and amyloid. So this is a study doing neurophysiology, cognitive studies, and pathology. So in this particular model, the brain shows extensive amyloid deposition, there's cell injury, there's learning and cognitive problems, and there's abnormal electrophysiology. The cells are hyperexcitable. So this shows a normal brain. These dots are hyperexcitable cells by the criteria used, and all the red dots are. And after treatment of beta secretase inhibitor, all this goes away. It really does a good job. And clinical studies, just looking at cognitive function on a maze and a platform, cognitive function improves. Time to do the platform task gets better. The amyloid deposition gets better. So this is a wonderful study. It shows a really dramatic effect in every possible realm: neurophysiology, pathology, and behavior. Yet, beta secretase trials have not been very effective. And so why? Well, first of all, beta secretase has a lot of functions. It's not there just to produce Alzheimer's disease, right? It affects sodium channels. It affects neuroregulin, which affects myelin. It's involved in photoreceptors, has effects on the muscle spindles, many other things. So it's a very important enzyme. If you just fool with that enzyme in a few months of an animal study, maybe you won't notice the difference. But in a complex human, you will notice that in terms of side effects that may not show up in the animal studies. And so that's very important that you're tinkering, if you tinker with this, something that affects many other things. Tau, of course, is the other hypothesis, and of course that's intracellular and aggregation. So let's go back and think. We need more money for research. Okay. We need the research focused on cure and prevention, not just behavioral therapy. Animal studies are important, but they have to be driven by clinical information that's accurate. We need animal studies and basic science to find better ways to make the diagnosis. So these things have to intertwine much better than what they do now. We can't do this with, here's a clinician group, here's a basic science group, oh, there are 5 really wonderful groups throughout the country. You need to bring so many people together to make all these things work. So here's some suggestions. Some of these are in conjunction with the Alzheimer's Foundation of America, which has made some real help in this. Increase funding. There's some move afoot by the Alzheimer's Foundation for the Check Dementia Now initiative, which will allow people in their tax returns to check a box. Remember in the old days we checked a box for presidential, to support presidential elections. There's a move afoot to allow people to donate when they put your tax return in. There's already some congressional interest in this, and we have to see how that's going. Make the clinical studies simpler. When you go to the DMV, you sign a donor card that says, if I'm in an accident, I donate my kidneys or my heart. Why can't we say I'd like to be involved in a clinical trial if such comes up? DMV is already collecting information about dementia because, at least in different states, you're not allowed to get a license if you have dementia. We could do a lot of this stuff at the governmental level to make the research and the populations much better. Add dementia to the census. Have anyone looked at the census questions? It says, by reason of mental, physical problem, is this person unable to walk up and down the steps? Does the person have hypertension? Do they have diabetes? Doesn't ask if you have Alzheimer's disease or dementia. You could ask that question. There are many other government studies out there that could be modified slightly to get more information. They're certainly doing this with some of these, but we're not there yet. If we could encourage them to do this, it would make a difference. We need to have partnerships. We've got to have global partnerships between the government, the clinicians, the drug companies, the universities, the advocacy foundations. It can't be that drug company X is doing a drug and they're going to make $1 million, and that research lab B got $1 million to study Alzheimer's disease. No, it has to be that we're all working together, and the drug company, university, we're all doing this together. We are not going to beat this problem because it's too difficult working alone. This is why we need the war on dementia, same as the war on cancer. I mean, now that everyone has worked together on all those other things. Again, modifying HIPAA. You fix it so that we can be more effective about clinical trials. I want to put in a plug for something that I think should come somewhere along the line, the Human Genome Project instead of genome. If we know, there's a ton of information about each of us floating in the web now. Amazon knows what colors you like, what foods you eat, how quickly you can push a button, how you decide between this and that. Tesla knows, do you turn the car to the left or to the right? What kind of decision making you make. All this stuff is out there, right? They have all this. They can use this information. So why don't we hash it so it can't be identified, store it in a big database? The NSA seems to have the capability to do that. So why don't we use those capabilities for positive good? Hash all this data so it can't be identified again and do correlations on this. It exists, it's there. The companies would love it because they could get a tax rebate for doing this. It's a great way. And why that's so important is there's a lot of things we all do. Do we visit a toxic waste dump? My iPhone knows if I've been near where a toxic waste dump is because it keeps track of wherever I've been. So we could find a lot of things about exposure, diet. Then if we take the genetics of people, we know their genes. We know what medicines they take. Great. We can put this together and really answer questions. And why that's important, there's a recent study from Lancet talking about the incidence of Alzheimer's disease throughout the world. And the idea of curing Alzheimer's disease is already done, right? Here's Turkey, 3 times the incidence of Alzheimer's disease as anywhere else. Brazil, very high. Argentina, low. Why? If we knew that, if we knew all about the people in these areas, we would be able to then say, you know what? Here are some more things to look at. Then go back to the basic science trials. Find out, okay, is there mechanistic support for this? Then go back to these kinds of demographic, clinical correlations and say, okay, do we find it there? Now, as a group, we've got a chance to do this. The experiment's already done. If I cut the incidence of Alzheimer's disease by a factor of 3 in the United States, people would say, fantastic, it's already been cut. So the answers are out there, just like we know that a lot of our developments came from natural experiments in the old days. We have to work that with our high technology in forming drugs, in doing clinical trials, and doing basic science. So there's a lot of things that we can do if we're energized and we're working together. So I think that's all I wanted to say except for that. And you know, my hope is that that is a sunrise, and I think it is.
Questions at the end.
Oh, we're going to take questions at the end.
Unless there's a burning question, we're going to hold off and do a little panel at the end.
It's this one.
Yes.
Okay, hold on. Yeah.
Great.
Okay.
Oh, that's the end. That's the last slide.
How do we get to the first slide? Sorry for my technical difficulty.
Okay, there's
Okay.
going to be a test now on that. I'd like, by a show of hands, how many of you have been touched by Alzheimer's disease in your life, by family or friends, by people you know? I guess I'm not surprised to see that. So I think everybody in this room has some personal feeling about needing to do something about this terrible scourge. And I know I feel very much that way. So my talk is Alzheimer's disease, many failed trials. Where do we go from here? And I'm going to go to some of the pathophysiology of Alzheimer's disease from bench to bedside. My background is as a clinician in internal medicine and as a molecular biologist doing research in Alzheimer's disease and cardiovascular disease at NYU Winthrop Hospital. And we are trying to look at the Alzheimer's pathophysiology using all human systems. And I stress that because of what we're seeing when we try to work from animal to human. And I'm not underselling the importance of the mouse studies and the other animal studies that we've done. But unfortunately, we've come up against a number of dead ends. And this one just came out in such a timely fashion right before this talk. And I emailed both of my co-speakers here today and said, I'm going to start with this slide. And this is the headline right out of the newspapers. Biogen and its partner Eisai confirm a decision to stop the phase 3 development of aducanumab, a potential treatment for Alzheimer's, a key part of the former's pipeline. So another drug that worked great in the mouse and doesn't work in people. And so there's a disconnect here. We need to put these pieces together to stop squandering money that could be used to find something that is innovative and different. And I believe in my heart that there is a cure out there. There is a way to slow this disease. The answers are there. We have to find them. We have brilliant minds working on it, and we can do this. So this is just a slide of the normal versus Alzheimer's brain, the plaques and the tangles that are the fundamental way that Alzheimer's manifests that we can see under the microscope. The pathology is the plaques comprised of the fibrillar A beta, the neurofibrillary tangles, and the toxicity of A beta that is thought to contribute by triggering pathological events that culminate in death of neurons. But whatever way you look at it, it's death of neurons. We have to find a way to keep the neurons alive, because that is what our brains need. We can't lose our neurons or our brains will not function well. It's pretty much fundamental at that level. And we know that cutting the amyloid precursor protein molecule in the destructive way leads to the amyloid accumulation. We also know that the APP molecule does have important function. It's not just a destructive molecule. What do we do to treat Alzheimer's? Right now, we have only a modest symptomatic relief for between 6 and 18 months. In theory, A beta removal or interference with aggregation will improve the signs and symptoms of Alzheimer's disease. But unfortunately, again, it worked in mice, but for reasons that we really have to figure out, it doesn't work in people. Acetylcholine is a critical neurotransmitter in forming, retrieving memories, and maintaining attention. And so cholinesterase inhibitors that slow the breakdown of acetylcholine are used to treat Alzheimer's disease. But the major problems, the mouse studies succeed and the human trials fail. Multiple attempts to use anti-amyloid antibodies by drug companies have not worked, and some of these companies have given up rather than trying to go in another direction. We can't predict who will develop Alzheimer's disease. We may have some genetic forms and some situations where we are aware that family members are increased risk, but we don't have any good prediction. And we don't have a way to test the treatments to see if they work, other than, as Dr. Stecker mentioned, 10 years of giving it to people or trying the animals. And we have a lack of direct access to neurons in the brain. Where are we with prediction? We don't have reliable biomarkers to predict. We have imaging that may be helpful, but a lot of times by the time you image it, it's pretty far along. We're starting to look at the abnormal microRNAs in the spinal fluid and other markers in spinal fluid. The new things I'm going to talk about today are neurally derived exosomes and models of Alzheimer's disease with brain cells from humans. So I'm going to talk about the way that we're going in the laboratory at my institution, and this is work that is being done in collaboration with both Dr. Willozin and Dr. Stecker. So how do we study mechanisms and test treatments? The model we're looking at is all human. It is human neuroprogenitor cells from Alzheimer's disease patients that behave like neurons. This is as close as we can really come now because you can't do a brain biopsy, right? It's not practical to do brain biopsies and grow human brain cells in a dish. If we could, that would help a lot. But we can approximate neurons from the brain. And we have ApoE4, presenilin-1, and non-AD controls, and we grow, maintain, and study them in our lab. And here they are. And I think you can even see that they look different. Even on the dish, the normal and the Alzheimer's look different. And I will tell you that these cells are incredibly expensive. So we eke out every experiment that we can with these cells. And I can say that our initial studies show even in just these neurons without any manipulation, synaptophysin, a protein involved in nerve communication that's decreased in Alzheimer's, and LRP, a protein that binds amyloid, are both significantly expressed differently to a lesser extent in the Alzheimer's disease neurons than in the control. So right away, there's physiological differences in Alzheimer's disease neurons. And I think we need to leverage that. We need to grasp that and figure out why and how and what to do about it. One of the things that is involved in this theory is that there's differences in the mitochondrial bioenergetics in these Alzheimer's neurons. The HNPCs from Alzheimer's subjects respond differently to high glucose than the HNPC from non-AD subjects. We want to study these abnormalities in mitochondrial structure and function in AD, and we're just starting to try to do that. Again, very costly. But what I can show you is if you expose the AD HNPCs to high glucose, they increase that base 1 that cuts APP in the wrong way much more than healthy human neurons do. So glucose, again, driver of metabolic function, and also I think the intersection of Alzheimer's and diabetes, where you're seeing that increased risk of Alzheimer's in diabetic patients. I think it's an energetic issue that we're starting to look at. And again, the high glucose also increases that precursor protein much more in the AD cells than in the normal. The NAHG is the normal cells. So two crucial functions of these neurons that are impacted by sugar differentially in the Alzheimer's neurons. And I think we're getting some clues. We're starting to get some clues. Now, obviously, neurons do not live alone. If you want to try to put a brain model together, you need the microglia, because inflammation, I think, is undoubtedly part of the process of Alzheimer's disease. And Dr. Willozin was really one of the pioneers in that area, that we have to consider immunity and we have to consider inflammation. And with injury, the microglia proliferate, transform into active brain macrophages, reactive microglia, and we know that amyloid activates microglia. So in our model, we want to look at neurons and microglia together. And so we want to add the microglia to the mix. We can incubate the microglia in a diabetic or a high glucose or a control environment. We can then take the incubation medium from the microglia and put it on the neurons to mimic interactions in the brain and see what happens. And I'll tell you, we were pretty surprised at what happened. Basically, the control has a low expression of APP. Whatever you do that mimics diabetes, because when you think about diabetes, what happens? The patient's sugar goes way up. They take insulin, the sugar goes plunging down. Sometimes they take too much insulin and it goes way too far down. Then they have a high level of insulin because they've taken insulin. Then they eat sugar and the sugar goes up. So this bouncing around of blood sugar and insulin levels, I think, is extremely damaging to the neuron. And I think that's manifested somewhat in this slide, showing that low glucose, high insulin, any of these combinations is going to increase APP. And that is probably because the microglia are being stimulated in an inflammatory fashion, and that is then affecting the neurons that are exposed to the microglial conditioned media. So kind of a clue here, again, where we want to use this information. So we can take neurons that we have made as HNPCs from Alzheimer's patients and non-Alzheimer's patients, but they're not your personal neurons. What if I want to do personalized medicine the way that we do for cancer? How can I do that? Well, there's a key part of the Alzheimer's machinery that we have easy access to. Even Kim Kardashian likes this stuff. We can take the platelet-rich plasma from anybody very easily, and platelet-rich plasma has platelets. The unique thing about platelets is they have the same Alzheimer's processing machinery as your neurons, but we don't have to go to your brain to get them. So we can access, through your platelets, the machinery that is processing amyloid that is similar to what is in your brain. It's unique to you. And I think we can develop a personalized approach to use your platelet-rich plasma, take your platelets, and tell you about your future risk of Alzheimer's and tell you how your machinery is functioning. And as I said, it's pretty easy. You know, we do this for sports stars to fix their injuries. We give them their platelet-rich plasma so we can get those platelets, and then we can study them. We can take the platelet-rich plasma from AD patients, and it's non-invasive. We can add it to our neurons of AD or healthy controls with and without the microglia, and we can look at the effects on the cell machinery, cell death, mitochondria, and we can see if treatments improve the function. We can do this right in a Petri dish, in our cell culture dish. We can add these different treatments and kind of give them a little first run. Our study is currently enrolling. We're studying male and female aged 65 and over. They're screened with neurologic history, examination, and activities of daily living with the Montreal Cognitive Assessment. We exclude if they have imaging and other studies that suggest another diagnosis. We have controls over age 65 who are cognitively normal. We do neurological history and exam to make sure that they don't have another cognitive disorder, and they have a MoCA above 26. We get their blood work, and we get their blood for their platelets, and we are doing this study right now, and it's difficult because you need the consent mostly of family, and a lot of times the family is supportive, but this is the kind of thing that we're doing right now on a small scale in New York and hopefully on a bigger scale. So we're able to take platelet-rich and platelet-poor plasma, because the platelet-poor is the control, because if we're correct, then the properties will reside in the platelet-rich plasma, and add it to our cells. What can we learn? We can compare the effect of platelet-rich plasma from Alzheimer's and controls. We can get a personalized profile of the amyloid processing of each patient. We can follow over time to see if we can predict those at risk. We can evaluate therapies as they develop, and we can correlate to changes with imaging and to other disorders such as diabetes. So our initial study, and I'll show this in two ways, is very clear that the effects reside in the platelet-rich plasma. And I can show that again. The amyloidogenic properties are only in the platelet-rich plasma and not in the platelet-poor. We get, with the platelet-rich plasma, we get a decrease in atom 10, which cuts the amyloid in a good way, the APP, and an increase in base. And these are SHSY5Y cells. So platelet-poor plasma had no effect. Platelet-rich plasma, as expected, had a significant effect. The other aspect of our study that we're working on is the neuronally derived exosomes, because the neuronally derived exosomes are a little bit of a way to get to the neuron without going into the brain, a second way besides the platelet-rich plasma. Exosomes are small extracellular vesicles shed from all cells. They have the protein, mRNA transcripts, and microRNAs from the cell. They're a fundamental mechanism of communication in the nervous system and allow bidirectional signaling. The microRNAs within exosomes can transfer between the neurons and the microglia and influence their phenotype, and they can affect genes involved in A-beta generation. So what we were able to do was look at APP, atom 10 base, and acetylcholine expression in neurons exposed to exosomes from control or AD plasma, and they're different. Again, we're just starting this. This is not published data. I'm showing you like our cutting edge work that we're kind of working as we get funds to do it. And I think that we have at least a start here suggesting something different. So what do we want to do with this? We want to get a predictor of who's susceptible based on their exosomes, based on their platelet-rich plasma. We want to leverage the similarity in A-beta processing between neurons and platelets to develop a test system. The advantage of this is all human components, it's non-invasive, it's neurons without a brain biopsy, and it is personalized. And we want to use the system to test drugs that have promise for AD treatment. So, what are we going to do? Look at the differences in the repertoire of microRNAs in neuron-derived exosomes from AD and control subjects. Pathway analysis to identify the canonical pathways that will be altered in AD based on the projected mRNA targets for the microRNAs that we find. And build on our model and test identified exosomes in the PRP-HNPC system over-underexpress the exosomes of interest in the system with mimics and inhibitors. The goal, treatments that are efficacious in humans, always the goal. And to prevent this that we see here, the severe Alzheimer's brain. So what are the next steps? We're looking at some off-label drugs that are being used. Nilotinib, a leukemia drug repurposed to treat AD, can penetrate the brain and is being used by some off-label. Litaglutide, a T2 diabetes drug, glucagon-like peptide 1 receptor agonist. Studies in AD patients with these drugs have been initiated by the National Institute on Aging and the Imperial College London, respectively, and you'll have to stay tuned for what we see. In the meantime, a little bit of practical advice. There were no targeted dementia prevention initiatives in the USA. Help us enroll patients and controls in our study. Contact our lab, become involved. Steps to take now: encourage your patients to eat a high-quality diet, engage in regular physical activity, increase social connections and intellectual activities, avoid head trauma, minimize heart disease risk factors. They seem very basic things, but so many of us don't do them, so that's why I say that to you today. And I want to acknowledge the incredible people who have participated in this work from my lab. I couldn't do that without this wonderful village. Laura, especially my postdoc, who did a lot of this work. I wish she could be here today. And Heather Renner, my tech, Dr. Gomelin and Dr. Pinkasoff, who are the clinicians that get patients to my lab, and Dr. Stecker. And I'm so grateful to the Alzheimer's Foundation and to the people there who have helped us to do this work. And also, I want to say just a word in memory of an incredible woman, Marianne Ragona, who passed away recently, and she gave funding to our work and helped to support our fellows, and she was a force to be reckoned with, and she had cancer and passed, and I miss her every day because she fought so hard for this. And I thank you for your attention. And now our third speaker of the day, Dr. Benjamin Willozin, and I'm so proud to introduce him and to have brought him here from Boston by way of Portugal. Yes, so now Dr. Benjamin Willozin.
Okay, well, Allison, thank you for giving me the opportunity to present here. I'm going to be discussing biomarkers and of Alzheimer's disease and new therapeutics. I'm going to first start this talk by covering about a general view of the field and then go into some of the work that myself and my company are doing. And that brings up a point, which is that I have a conflict of interest. I started a company, Aquina Pharmaceuticals, and I'm the chief scientific officer for that company. So when we think about Alzheimer's disease, the first thing that's become evident from all the studies is that the disease process begins up to 20 years before we actually see behavioral changes in patients. So that presents a challenge for any kind of development of therapeutics, because a lot of the changes, key changes, might have already occurred by then. And in particular, when we think about amyloid, there's a general consensus that amyloid is an important contributor to the disease process, mainly because of the genetics, and it's the classic for the definition of pathology. But it may well be that since amyloid is outside the neuron, once it has already turned on the process in the neuron, removing the amyloid outside the neuron is insufficient to prevent disease progression. That's the way people are starting to think about this. So that's the first challenge of facing disease, that by the time grandma shows up at the clinic, there's already a lot of damage in her brain. Second major problem, which Mark's pointed to, is the heterogeneity of disease. When we talk about Alzheimer's, really what we're talking about is dementia. And there are many, many different types of dementia. Alzheimer's type dementia only constitutes about half of the cases of dementia. Other major elements are vascular dementia. So vascular disease, even among patients with Alzheimer's disease, vascular disease is a major contributing factor to the process of cognitive loss. And then you have other mechanisms of neurodegeneration that occur. You have Parkinson's disease, which is driven by alpha-synucleinopathies. And classic in here is diffuse Lewy body disease, which gives a dementia, a different flavor than the dementia of Alzheimer's, but nevertheless a dementia. You have amyotrophic lateral sclerosis, which we mostly think of as a motor neuron disease, but the more they've studied it, the more they realize that there are significant cognitive deficits associated with ALS also in many but not all patients. And then there's the frontotemporal lobar dementias, which includes FTDP17, PSP, CBD, PICS. Those are these classic dementias that affect the frontal part of the brain that can also appear as a dementia and without careful analysis be misdiagnosed as Alzheimer's disease. And then we're aware of these things called PARC and RTAG and PART, which are like tangle only, amyloid only, all these different variants. And then what's received a lot of attention recently is chronic traumatic encephalopathy that's associated with sports injuries and military service. So we've got this heterogeneity of disease that we have to deal with. And the other thing that I've already mentioned, but I want to emphasize in the next two slides, is the contributions of metabolic and cardiovascular disease to cognitive function. So anytime you treat a patient trying to address cognitive function, if you don't deal with the underlying metabolic and cardiovascular problems, you may well be missing a major aspect of disease. So here's work done by Mia Kiva-Pelto showing that looking at each of these cardiovascular risk factors, mass index, systolic blood pressure, cholesterol, at midlife. Okay, so this is stuff that we're here. Some of us are at midlife. These are already factors. And you can see how with each increasing risk factor, the risk of Alzheimer's goes up many-fold, up to six-fold, and diabetes itself increases risk six-fold. Perhaps not surprisingly, epidemiological studies and some prospective clinical trials are suggesting that if you treat blood pressure in a preventive manner, you can actually reduce the risk of Alzheimer's. But that doesn't help grandma or the person who shows up with cognitive loss. By then, it's too late. Loss of cerebrovascular function, loss of blood flow, was one of the first things noted in the disease process. So this is one of the first things that can be seen in patients with Alzheimer's disease, is that there's a loss of cerebrovascular blood flow. And in a very simple way, if you think about this, it's easy to understand. No blood to the brain, you're not going to think well. Okay? But it's even more complicated than that, because anytime we utilize a different part of our brain to engage and have a cognitive function, we shunt blood there. So it's not simply how much blood is flowing, but it's whether our neuronal systems are reactive to that. So people are beginning to look at the reactivity of the neuronal systems and responsivity to develop tools to improve responsive blood flow in the brain. And there's a lot of work going on there. What can we do about it? Well, Allison mentioned this, and this is really the striking data we have so far. I must say, having worked in the field for 30 or 35 years, I find it depressing to say that after tens of billions of dollars spent in the field of Alzheimer's research, all we can say is eat well and exercise. But that's where we're at, and the data are cogent, so I thought I could show it to you. So these are two different studies, one led by Mia Kiva-Pelto and the other led by Tobias Hartman. And this is work from Lancet Neurology, from Tobias's work. And what you can see is this is the kind of total neuropsychological test battery, and this is the CDR score. And these are patients who were given a paradigm of exercise and a nutritional intake that promotes development of lipids necessary for the brain. And they did this in a preventive manner, looked at it prospectively, and what they found, and just so you get a sense, the control population were people who were kind of given supervision, given equal amount of clinical care, but not given the nutritional supplements and not told really specific exercises, but they're given a lot of psychosocial care. So they get not as much human contact. And what you can see is the dramatic lowering of disease progression among patients who received the kind of intensive therapy that was given. And this has been shown now in multiple studies. So just like with cardiovascular disease, there's no question that anything that improves our general health, exercise, eating well, improves the disease process. And I was talking with Tobias, one thing to be realized, even though this nutritional supplement promotes lipid synthesis in the brain, it actually does not increase cholesterol levels. So it's actually good for cardiovascular disease too. So the reason I present this, and I'll also present a couple other things, is to give you these different kind of non-classic non-classic therapeutic ways of addressing disease in line with the general thinking of this symposium. So the next focus in the field has been on biomarkers. And the reason why biomarkers are important is because they enable three things. First, they enable precision in cohort selection. Imagine you're starting a clinical trial and you want to do a drug that lowers amyloid, and you take 100 patients. It turns out 30 of them don't even have any amyloid in the brain. Well, those were the problems that plagued early clinical trials. And so we realized that whatever you are targeting in Alzheimer's, because it's a heterogeneous disease, you better damn well make sure that the patients you're putting into the trial actually have the marker that you're looking for to reduce. Because otherwise, right off the bat, your study is going to be underpowered and you will fail even if your medicine works well. Second point, evaluation of target engagement. If you have a negative trial, is the trial negative because the drug was ineffective or because the drug didn't get into the brain or last there long enough or have enough free compound available in the brain to engage the target? So in order to interpret a negative trial, in order to be confident that you're using sufficient dosage to engage your target, you need to have markers of target engagement. So that's been a key thing that's been happening that people are starting to do in the trial, but that isn't always possible, which creates trouble. Then the final thing for biomarkers is sensitivity. If you switch from a subjective human scale, like cognitive measurement, to a biomarker, you reduce the number of patients needed in a trial tenfold, or you increase the power or the sensitivity of your trial multiple-fold. So by switching from subjective commentary to biomarkers, you increase the sensitivity to detect outcomes. Considering this, in 2018, the NIA came out with a new labeling system for Alzheimer's disease. And it's called the ATN system, and you can expand it even to be the ATNC system, which A stands for amyloid, T stands for tau, N stands for neurodegeneration, which means MRI, and C is cognition. So I wanted to show you how that works. It becomes important in evaluating cases so that you don't just say Alzheimer's disease. You say, this is what this patient is like. So here's an example. Here's a classic case of Alzheimer's disease, and this was, Bill Klunk showed this. You can see, this one actually shows you how powerful the imaging is. So if it used to be that we would just use MRI or CAT scans to look at cognitive change, to look at neurodegeneration. But the problem is that the changes, people are variable, they're heterogeneous, and it's very difficult to look at an MRI and have a definitive answer unless it's very clear. But when Bill Klunk discovered the Pittsburgh B compound, and now they have similar compounds for tau, which I'll show you in a moment, you can see that there's this dramatic change that you see in patients who have Alzheimer's disease. Control patient, Alzheimer's patient, it lights up. Compare that to MRI, where you don't see, the difference is not as evident, it's easy to make mistakes and misdiagnose. And this is FDG, which is measuring glucose in the brain. Again, there's a difference, but difficult to see. Once you can see the amyloid, I'm just putting this up here as eye candy, apologies, but you can kind of see what the distribution is in the brain. So here you can see the brain from all sides, and you can see where the amyloid is accumulating in this characteristic case. Now, so that now we can test for amyloid accumulation in the brain. I'm not showing you, but we also can test for tau accumulation in the brain. We can use MRI, and we can start to develop these ATN and add the C component to it to look at diagnoses. So here's some examples. Here's a classic patient, which is A positive, T positive, N positive. So there's the amyloid. There you can see the tau, and you can see the massive degeneration, very clear. Here's a different type of patient. This is amyloid positive, but now they're tau negative, and they're MRI negative. So this is a type of patient who could be either of two possibilities. Either they are very early in the, three possibilities. One, they're very early in the disease process. They have the very first thing that occurs, which is amyloid, but they don't yet have the degeneration of tau or degeneration seen by MRI. Other possibilities are that this is someone who perhaps is not going to develop degeneration. 30% of elderly patients have amyloid but no degeneration. And finally, as I'll show you in a little bit, this could be a patient who has a different type of pathology. And so here's an example of a different type of patient. Here's a patient, they're amyloid positive, whopping amounts of amyloid, but they're tau negative, but they have lots of degeneration. So what is this? Well, when we think of Alzheimer's, we say amyloid and tau, but those aren't the only pathologies. There can be TDP-43 accumulating in the brain. There can also be synuclein. And this is actually classic for dementia with Lewy body disease. So once we have markers for synuclein, we can add that to the mix. They're not quite there yet. So those are the clinical biomarkers, the imaging biomarkers. Now we can move on and say, what about biomarkers of blood or CSF? And these are the general areas that are being looked at. There are the kind of classic pathologies of amyloid and tau, phospho-tau. There's inflammation, where the markers appear to focus, on innate immunity, not the adaptive system of antibodies, but the innate immunity that respond to like neurons suddenly changing their lipid profile on their membrane surface. And then we look at markers of synaptic dysfunction and axonal damage. So when we look at this, we have two choices. One is CSF, the other is serum. You can see when you look at CSF, there are a number of markers that work quite well, including the most promising are the tau and neurofilament light chain. Neurofilament light chain, in general, appears to work for almost every neurodegenerative disease. As neurodegeneration increases, there's more neurofilament in the CSF, and it seems to be very consistent with the disease progression, and also, at least in cases where there's one disease, SMA, where they have an effective treatment, and as you give an effective treatment, the neurofilament goes down. So now I can tell you, as we think about clinical trials, neurofilament is the go-to measure to look at disease progression. But a lot of patients are hesitant about having lumbar punctures. That's characteristic of US. That's also characteristic of Southern Europe. Interestingly, in Nordic countries, whenever you go to a neurologist, they have an LP. It's not considered anything special. So there's a cultural component, but it's a very real component. So people are looking at serum. In serum, tau appears promising, particularly in chronic traumatic encephalopathy. Elevated tau is pathognomonic for the disease. And then inflammatory signals also appear to be promising. These are the ones that in meta-analysis are holding up. So where does that leave us for targeting drugs? We can think about this in several areas. First of all, let me just go back one moment. Classically, people have looked at post-translational modifications, phosphorylation of tau. That so far hasn't worked because when you target kinases, they have broad actions that seem to target too many things, so they fail due to side effects in clinical trials. What's been incredibly promising are the vaccines. So as Allison mentioned, the vaccines for amyloid failed, hugely disappointing, but there are vaccines going through for tau and synuclein. And also one, you might not know this, but something called C9orf72. So these are now in phase 2 clinical trials, and I predict in the next 2 or 3 years we'll be hearing results from those. And if you imagine that the reason amyloid failed is because amyloid is outside the neuron and too early in the process, if you target things that are in the neuron, you might imagine that there's a better chance for success here. So that's the thinking. We'll see. Another thing that people are doing are antisense oligomers to lower tau, lower synuclein. There's a lot of enthusiasm because of you can knock out tau, knock out synuclein, and the animals appear fine. I personally find it difficult to believe that knocking out, getting rid of tau won't be a problem. And in fact, there are a couple conditional knockout models for tau that have been made that show that if you knock out tau as an adult, there actually are cognitive changes that occur with time. So we have to wait and see how that is going to work. But what you should get in your head is antisense oligomers. Those are an effective strategy that has been clinically proven to work in humans to modify disease course, and so the field is strongly focusing on antisense oligomers. The other thing people are focusing on is AAV. The difference is this is reversible, so if your treatment is bad, you can stop it. If you give an AAV and your treatment is bad, you're stuck. Finally, there's what the company I started, and I'm going to talk about in the upcoming slides, is looking at stress granules. And I'll tell you what those are in a moment, but those are expected to block the pathophysiology in the process of both forming pathology and also what I've shown recently is they also they also increase removal and they reduce spreading. So that's why I'm so enthusiastic, but time will tell. So Allison mentioned the Biogen trial failed. You might criticize Biogen, how could they be so stupid? Well, this is the answer. Look at the amyloid. The antibody totally cleared the amyloid, okay? There is every reason to imagine that if amyloid was important, this drug would do it, okay? Would be preventive. It's an incredibly potent drug, but removing amyloid appears to be insufficient to block disease progression. So what else can we go to? Well, I thought I would give you a couple innovative therapies before I went into the stuff I'm doing. So Li Wei Tsai has shown that if you use 40 megahertz frequency in the brain, it activates microglia and helps remove pathology. That's good. Caveat? Caveat is it only works in the sensory input that you're using. So you do vision, it's going to affect the visual areas, but other areas it's not going to do it. Sensory, same thing. So we need to see, and this was looked at for amyloid, hasn't been looked at for tau, extracellular versus intracellular. So we'll see. Another example, this just came out, Ken Kosik's lab used farnesyl transferase inhibitors, which were originally developed for cancer. So those look promising, that it reduces the accumulation, in this case, of tangles, so that's great. However, it only works preventively. They tried it after pathology was there, and it doesn't increase removal of tangles, just formation. That doesn't help once you already have the disease process. So enter the stuff that I do and has really taken over, starting with the field of ALS and now spreading to Alzheimer's disease. This story is an interesting story because it conveys a biological mechanism that we didn't have when we learned about how the cell works. And it's called liquid-liquid phase separation. So I don't know about you, but when I think of a cell and I think of all the thousands of molecules floating around, I find it hard to believe that they really can find each other. It boggles the mind. But what a number of people have found, this was led originally by Alan Rosen, and then Mark Rosen, and then Cliff Brangwines really pushed this, Simon Alberti, but here you can see the basic idea. It turns out that proteins have these areas that are low affinity these areas that are low complexity domains that are a little bit sticky. They bind to each other, somewhat specifically with low affinity. And so it turns out when you put these types of proteins in solution under the right conditions, they form droplets. This is not lipid droplets. It looks like oil and vinegar, but these are actually protein droplets. They can fuse, they can separate, they look exactly like droplets. And they're associating due to a combination of biophysics, Gibbs free energy, and low affinity associations. The problem is this biology, which is fundamental to the way our cell works, also has a weakness, because when these things come together for a long time, they switch into a lower energy state, which is beta sheet and amyloids, and they develop, some proteins develop these irreversible amyloids that give rise to these disease features such as we see with TDP-43, which is in ALS and frontotemporal dementia, and neurofibrillary tangles, tau. So there are dozens of different types of RNA. So the way these work is, these became important, were first realized for RNA binding proteins. Because it turns out RNA is a scaffold that helps promote liquid-liquid phase separation. And RNA binding proteins control RNA metabolism through this process. If you think about RNA, you've never seen it move around in a membrane except for microRNA. You know, everything else, there's a membrane around the nucleus, membrane around the mitochondria, around the lysosome, around the cell. There should be a membrane around the White House. But there is, but you don't see membranes around RNA. The reason is because RNA binding proteins act by coalescing through phase separation and binding the RNA. This becomes particularly important in disease where you develop these things called stress granules. So let me explain this. What happens is, under normal circumstances, RNA binding proteins are localized in the nucleus, where they do RNA splicing, and in with disease, when you have stress, your body needs to take the proteins and adapt the protein synthesis to the stress. So what happens is the RNA binding proteins flood out of the nucleus, bind up all the specialized RNA that's not important for stress, allowing the cell to focus protein synthesis specifically on proteins related to stress. That's good. It's supposed to be in a acute transient phenomenon, but what happens is in diseases like Alzheimer's and ALS, it becomes chronic and persistent, and then you get those hairballs that I showed you in the last slide, and that's pathology. So the importance of this is it's biological. There are all these enzyme systems that are built to regulate this and can convert it to go from here and back. And so the idea that I had was that if we develop a high throughput screen, we can kind of in an unbiased manner select for compounds that can modify this biology and reverse it. And so what we've done at my company, Aquina, is we labeled TDP-43. We made cell lines that could really do that could do a high throughput screen. And the idea is you develop these cell lines, you get the conditions where you start to accumulate TDP-43 in the cytoplasm out here, and it's out of the nucleus, and you do the high throughput screen. And, this is just what it looks like. This is basal conditions, TDP in the nucleus. You give it a stress, it all goes out into the cytoplasm. And here's an example of how you can reverse the process. If you give cyclohexamide, even though there's a stress, it keeps it in the nucleus. I can explain afterwards why, but there's biological reasons why. The fact that it reverses it suggests maybe we can get non-toxic inhibitors to treat ALS. And in fact, we did that. We've developed a number of compounds that prevent and reverse stress granule formation. I think Allison saw a slide that I have to take out because this is being filmed, but you can put these nice compounds on and they just melt away the pathology. It's very nice, very promising. So what about Alzheimer's though? So the idea is let's apply this to Alzheimer's disease. Now, I came up with the idea that tau might be related to the stress granule pathway, because if you look at the genes that cause ALS and frontotemporal dementia, a lot of them are RNA binding proteins controlling RNA metabolism. Admittedly, some of them are controlling proteins degradation and others inflammation, but there's still a strong RNA metabolism component. So one wonders whether tau is in this continuum because it's participating in this pathway. And so early on, we showed that, in fact, it does. So if you take a cell and you put in tau and give it a stress and look at stress granule markers like TIA1 and PABP, you see formation of stress granules. And hard to see, but there's tau also forms in those TIA-positive stress granules. If you treat with arsenite, you can prevent this all. So that suggests in cell lines that there's some connection between tau, stress granules, RNA metabolism. We then went to primary neurons. We used optogenetics, where we took a TIA, we added a photoactivatable GFP to do a pulse chase. And you can see if you take a neuron and you pulse it, turn it to green, then follow it over 6 hours. After 6 hours, it has gone away due to a mixture of degradation and dispersion. But if you induce stress granules, as you can see here, it's completely stable. And in fact, we showed not only is it stable, the tau that's in those granules has started to aggregate. So now you have a direct link between stress granules and tau aggregation. And when you go and look at the brains of patients with Alzheimer's disease or frontotemporal dementia, you can see that wherever you see green is tau pathology, red is TIA pathology, the two co-localize. But it's not true of all RNA binding proteins, like G3BP, a different one, doesn't co-localize. Now, I don't have time to go into this, but there's a lot of technology here that it turns out that RNA binding proteins are highly sensitive to fixation methods, and we have a publication in 2018 that discusses that. So, what's the idea? The idea here is that in stress in neurons, what happens is you get these stress granules that form, and tau co-localizes with the stress granules. But then with time, it becomes, you get persistence, and with that, the tau kind of gets its pathological changes and becomes persistent. Then even if you remove the stress, this stuff stays as the pathology that we ultimately see. To determine whether RNA binding proteins were relevant to tau biology in vivo, we did a in vivo test where we took a classic model of tauopathy, a PS19 mouse. We crossed it and reduced TIA by 50%, and then looked to see what happened. We looked at a number of different ways. We looked at synaptic fields. We looked at cortical thickness. And here we've looked at neuronal number using Nissl or NuN. And what you can see is that this is a P301S mouse where you see a loss of the hippocampal neurons. And here, when you cross it with TIA, reduce TIA by 50%, you have a rescue of that. And no matter how we looked at it, we saw a rescue of neurodegeneration. And if you look at survival, you see this striking rescue here, dose-dependent, depending on the amount of TIA that you've reduced. So that says that reducing TIA can prevent tau pathology. But what about other mechanisms? And in particular, one of the things we've become aware of in the field is that not only can tau be induced stochastically, but pathology can also spread. So we recently published a paper where we did the same thing, where we took TIA het mice and crossed them and injected oligomeric tau. And then we looked in the ipsilateral and contralateral areas. And what you can see is when we injected oligomeric tau, we also injected fibrillar tau and compared them. You could see that the oligomeric tau was very toxic, whereas the fibrillar tau was not at all toxic, which is consistent with what other people in the field have shown. When we looked at the ability of TIA to impact on that, if we reduced TIA by 50%, we could prevent the spread of the tau. This is the oligomeric tau shown with TOMA2. We could prevent pathological stress granules and in protect the animals. Interestingly, it only worked against oligomeric tau. It didn't do anything for the fibrillar tau, showing that not all things are the same. So that says that this potentially has viability as a mechanism to prevent disease propagation. So we went and did the same thing I told you about for TDP. We developed a cell line that expressed TIA and tau, did a high throughput screen, and developed a number of currently going through compounds that we have identified that can prevent this and also reverse it. And so this just gives you a sense of what we have in place. So here's the development of stress granules over time in SY5Y cells. And this is just shown for TIA, but wherever you see TIA, you see tau also. In this, TIA is green, TIA is red, tau is green. Then this is our algorithm for quantifying it. And you can see, very easy to quantify. And this would be a positive hit, which doesn't develop anything because it's preventing it. And then we do medchem and bring that down through. So I anticipate that we will be able to, that the company will be pushing this forward. Will it work? No idea. But this is a symposium on novel therapies. So I wanted to leave you with an overall way to think about how this integrates with the field. So we know there are a number of neurodegenerative diseases. There's ALS, there's tauopathies, and there's Alzheimer's disease. I want to submit to you that these are actually part of a similar pathway, and that the disease that we develop, that we diagnose with, depends on where you enter in the pathway. Alzheimer's disease, the diagnosis requires the presence of amyloid and tau pathology. So the only things that can be categorized as an Alzheimer disease gene must induce beta amyloid. So these factors induce beta amyloid, and then as they go down the pathway, they induce tau pathology, which mediates the disease. And then tau pathology, as I showed you, acts through RNA binding proteins to induce the disease. So for Alzheimer's, we are positive for amyloid, tangles, and RNA binding protein stress granules. Now, some mutations just induce tau, but this is a feed-forward system. Tau is in the neuron, it doesn't induce amyloid. Ergo, by definition, it does not cause Alzheimer's disease, but it goes through the same pathway and induces disease neurodegeneration through a pathway mediated by RNA binding proteins. And then finally, some genes just are RNA binding proteins, and they induce degeneration directly. The significance of this is that you can imagine that therapeutics that act here or here could act against here, whereas the therapeutic that here will not prevent this or this. Correspondingly, I haven't talked about TDP-43 much. It's associated with frontotemporal dementia. It seems to follow a similar pathway, but a different set of stress granules. So with that, I'll just acknowledge the people who have helped me, the team, the funding, and now we can go to questions.
So let's see. So we have time for some questions from the audience. If you want to ask, please go to the microphone.
Yes, there's a very unifying metabolic causes that have that weren't really mentioned here. That is in old age, aging brain, oxidative stress in particular is a cause. And oxidative stress inhibits methylation, and methylation of PP2A affects the phospho-tau level. It affects the APP phosphorylation. It affects alpha-synuclein. Methylation affects even your stress granule formation. And so it seems as though this metabolic factor that goes along with old age is an addressable therapeutic approach for neurodegenerative disorders. And in fact, there's been a number of studies suggesting that that might be the case. But it's not patentable. The big problem, it seems to me, in terms of moving that forward is the fact that it doesn't present a druggable, patentable target. And you have a lot of people with Alzheimer's out there who don't care about whether it's a patentable idea or not.
So it's not simply that it's not patentable. People have actually tried numerous trials of antioxidants for Alzheimer's, and they weren't successful.
Well, some have been successful. David Smith, Helga Repsum, for a perfect example, PNAS studies and things like that. So I wouldn't put a blanket
So
over
the
it
major
and say, yeah.
studies have not been successful. The other way, I acknowledge what you're saying, but the other reason that people haven't followed it as much is there was a Broad study, a study done from Broad Institute looking at the likelihood that a drug would make it through clinical trials. And the stronger the genetic relevance, the more likely the drug was to make it through. So a drug with high genetic relevance had a 70% chance, medium 40% chance, low genetic relevance, genetic validity, 10% chance. So that means those low genetic validities are the ones that make total sense, but actually there's no genes that have been shown to influence it. Let me finish.
I'm sure the Broad Institute was interested in a genomic prediction.
I'm just saying that the field has looked at the likelihood of success and taken that to heart. I'm not defending it. I'm just telling you that that's kind of, those things have led people to look at different foci. There is no question that epigenetics is very important, and lots of people are looking at that. So I agree with you on that.
Okay, thanks.
So my question is to Allison. It seemed that you were indicating that we need to go to a human environment because the clinical trials are failing. And my question is, do you have any evidence in that a mechanism or something is different in the humans? Maybe you showed it and I just didn't see it, different from the mouse. Because there's two ways that this could happen. One, the mouse model could be wrong, right? I mean, we could be assuming that beta amyloid and tangles are what are causing Alzheimer's disease, and it could be just not true. And then if you make a mouse model on that, and then it fails, it could be telling us that the whole premise, that beta amyloid is maybe not toxic, not the problem, which is what the data would suggest. And then if we abandon mice because the model failed, then we're really, instead of looking at the data and saying, wait a minute, maybe the whole model was wrong, and then we move on to human and keep assuming that that model's right, because there's both ways, right? It could either be that the model was wrong, the beta amyloid isn't the problem, or it could be that the mouse is different. And so my question is, do you have data that suggests that the mouse neuron really is different, and how can you make that judgment, and then and this is about moving forward, right? Do we really abandon it?
Yeah, it's kind of like I'm thinking in a more elementary level of what you're saying. So the mouse model works for the mouse, and the mouse neurocognitively does better when you make the change. But if it doesn't work the same in the human, you have to use the human system, which is not a model, but the reality, I guess,
that's
to
my
me.
question.
And
What is the evidence in the
my
human?
example to you would be the example of COX inhibitors. So COX inhibitors do not cause accelerated atherosclerosis or cardiovascular risk in mice. In fact, they're good for them because the inflammatory effects are dampened. But in humans, you have increased cardiovascular risk with COX inhibition. So humans and mice are not the same. I don't have the information to say exactly where the deviation
So
is.
nothing in your data showed that the
Well, my
the
data is
human
human
cells,
data. In
yeah,
human
but compared
cells,
to a human, did you have any data in human that if you went to a mouse cell would be different?
I didn't look at the mouse cell. I don't know about platelet-rich plasma in the mouse, so I can't really answer. I don't have that data.
You haven't looked, but wouldn't that be important to look to see, is it really different?
I think that it's more important to see if the system works.
I would just point out that we all have to work together on this stuff. That's the whole deal, right? You need mouse, you need rat, you need human, you need everything. You
I'm
need
not
biochemistry.
diminishing
Yeah.
that the mouse is valuable.
In terms of trying to increase the database for potential Alzheimer's disease patients, have you considered mining the NHANES database or trying to expand the NHANES to be more cognizant of Alzheimer's disease
Yeah.
risks?
Allison's been spending a lot of time looking at that. I started to, but a lot of it's not all that great. Allison, you've looked a little more into that than I have.
Yeah, we have done a little bit of work with NHANES. My postdoc is very good with biostatistics, and so she's interested in it. But she's doing it more for dietary effects, where they collected a little better data. The problem is a lot of the data is really that she has. It's older, and it's only kind of gives you suggestions and guidance where to go. But we definitely have talked about it.
I think there are a ton of other databases out there besides that. When you really look, there are hidden databases everywhere that the government collects. And if you could put them all together, you'd be in much better shape. Get your DMV, get your agricultural people together with the Census Bureau. All that gets a better collection. But yeah, NHANES will be a part of that for sure.
Hello, I have a question for Allison. Ivana Delala, Boston University neuropathologist there. In your experiment where you exposed neurons to exosomes from plasma, from AD and controls, but these are exosomes that are secreted by numerous organs, and one would argue that majority would come from like big organs, liver and so on, and that very few, or you have...
No, you have to take the neuronal derived exosomes.
How do you define...
It's one of the, it's CAM. Yeah, it's NCAM. It gets
But
adhered.
NCAM?
So it's specific. But you're right, I wouldn't want to do the whole
Okay,
exosome.
But,
uh,
but
repertoire.
if you do, maybe you know that if they would be coming from, and maybe they would then reflect metabolic signatures that would actually also affect APP processing in neurons, and there would be that connection between metabolism, diabetes, and AD that, you know, is reflected in this suggestion that eating well and exercising is actually helpful. So I'm glad that you have found a way to isolate brain-specific exosomes in periphery, because
Well, I can't take credit for it, but it is out there,
But
and that is, I think, very
well,
important to do.
Okay, then it'd be great to actually convince NIH reviewers that it's possible to find the brain-specific exosomes in periphery. But thank you for that. And also, I have a question for Ben. Thank you for this wonderful talk. Since you mentioned and emphasized, I think, very important point about stratifying patients that go into trials. And now that the human genome is actually not that expensive to obtain, are you also considering obtaining polygenic risk scores based on VAS and GVAS and categorize these participants even more so, especially considering that now we know that there's maybe 6 pathways now being potentially affected and would that actually be helpful in?
Yeah, so there's 2 strategies that companies are using depending on how big the company is. If you're a big company, you go directly into an Alzheimer group and you stratify, do genetics ahead of time, you stratify. What a lot of companies, including some of the big pharma, are doing, particularly with the tau therapeutics, are instead of going at Alzheimer's, they're actually going at progressive supranuclear palsy, which is an orphan disease, and it has a very rapid decline, and it has tau pathology as the pathology. So I know that's what my company is looking at. So it's kind of 2 different ways of addressing it, but there's zero question if you're going to look at Alzheimer's, you have to genetically stratify. Yeah.
Hey, my name is Luke. I just have a simple question. So at one point, did you look at cerebrovascular blood flow with Alzheimer's dementia?
We haven't done it, but it's been looked at since the 1990s.
Okay.
And it's strongly, I mean, it's just a very strong signature. What's your question?
Yeah, so my question is, obviously cerebrovascular blood flow correlates with CSF flow. Have you looked at, or has there been any thought into looking at a correlation between measuring intracranial pressure using ocular ultrasonography and the onset of Alzheimer's dementia?
So I'm not aware of that, but what people are looking at are glymphatics.
Okay.
Which are which kind of don't have their own flow, but they respond to the cerebrovascular flow, and in that case, what happens is, it turns out that as you get cardiovascular disease and you get arteriolar stiffening, the shape of the pressure wave changes from being a broad pressure wave to a sharp pressure wave, and it turns out that reduces glymphatic flow in the brain. So I don't think that directly addresses your question, but it does say, you know, it gives you a concrete idea on how cerebrovascular flow could directly impact on removal of waste measures from the brain. And I guess the reason I brought it up is that I know at BU there's a guy, David Boaz, who's using kind of Doppler imaging to look at air bubbles, micro air bubbles in the brain. And you can use that to directly measure cerebral vascular blood flow.
Thank you.
Hi, thank you for your talks. Just a quick question. Most of this seemed to be focused on amyloid and tau. Can you speak to the vascular hypothesis and maybe talk about that briefly and how it might contribute to Alzheimer's?
So first of all, there's cerebral amyloid angiopathy, which is a mutation in APP that causes an amyloidosis that occurs in the vasculature. So that's a direct link between amyloid and vasculature. The second important link between amyloid and the vasculature is that even in sporadic cases, you find amyloid accumulating both in the endothelial cells and in the smooth muscle cells around the vasculature. So the entire neurovascular unit is kind of messed up and dysfunctional. When people look at it, what they find is that it's mainly the arterioles, which have a smooth muscle, that are clearly affected in terms of pumping. But even if you look at the capillaries, what you find is that because of both amyloid and tau change the endothelial cells along the capillary, such that they become more adhesive towards lymphocytes. So you get a lot more lymphocytic adhesion in the capillaries, and you have a reduction in capillary flow. That's been shown in animal models. I don't think it's been shown yet in people. But that's a second hypothesis in there. And then there's some small number of papers that look at hypothalamus, which also controls the vasculature, which is affected in Alzheimer's. So the general hypothesis, I just say, is I don't think there's a specific answer to the cerebrovasculature in the brain, other than kind of the brain needs nutrients. I forget what percent of the body the brain weight is, small percent, but it consumes 20% of the energy.
Last question.
Hi, I have a question for Allison. Just as a disclaimer, I'm an undergrad, so I'm sorry if this question is way out in left field. But
No, never. Always ask.
I find it really interesting that you found some correlations between inflammation and diabetes to Alzheimer's. I was wondering if you have any ideas or thoughts about maybe the parallels between advanced glycation end products in Alzheimer's disease and how it affects the body in hyperglycemic conditions and diabetics.
Well, we've done a lot of work actually on the advanced glycation end products in the cardiovascular system. So I think it's multifaceted, and I hope that we're going to look at some of that in the brain, but certainly those products are very damaging to blood vessels, which is very bad for the brain in and of itself, on top of any other effects that might be there. And I think this is a very active area of investigation, because we're seeing more and more this overlap. And trying to figure out how to control it is very important. So thank you. Thank you all. Thank you so much. I think it went well. We got a really good attendance.